Now it's my pleasure, yes, thank you, to introduce Naomi Horiba, who is a medical officer at the FDA. You know, we've been partnering closely with the FDA over the past year, led by Mitesh Borad, as we think about how best to design clinical trials that are rigorous, efficient, and patient-centered. And they've been very receptive to our suggestions about companion diagnostics, clinical trial design, and we really look forward to your thoughts.
Thank you for saying that. Thanks to all of you for coming, and I want to thank the organizers and the Cholangiocarcinoma Foundation for inviting the FDA to this very important conference. On a personal note, I have had a very meaningful experience over the past few days here, and I think that it's important to have heard all kinds of stories, patient stories and scientific stories. But also, I hope you all know what a remarkable place this is. I don't think I have to tell you that. And I'm actually hoping that this will serve as a model for other patient advocacy organizations because it's truly a successful model. So we're going to change gears now a little bit, and I will be talking about some of the aspects of FDA that I think may be less well understood, and I'll be speaking a bit about some of the expedited programs. I'll touch on companion diagnostics. But let me first share with you what our mission is so you can kind of understand where we're coming from. Our mission is to serve the American public and to ensure that drugs are used safely and effectively. And that's pretty self-evident, but our mission also encompasses disseminating information to patients, and so we do this through the label, and so that provides a comprehensive and yet a concise, maybe not so concise if you've ever looked at a label, but it's a great way for us to communicate what we think is important based on the trials that have led to the marketing of the agent or device. The other aspect of our mission that we take seriously is the need to expedite getting drugs out to the public. So as you can imagine, there's some push and pull between those two aspects, trying to get drugs out quickly, but trying to define them well enough to be able to describe it for patients and for their providers. So I'm going to talk, everything I talk about here is going to be publicly available information. And really any specific questions or issues really should be directed to the appropriate center because I cannot possibly know everything about every scenario or device or drug. So this is a brief outline of my talk. I do want to highlight a bit about the Oncology Center for Excellence, talk about some of the efforts that have been undertaken to help facilitate getting drugs out in terms of novel trial designs, endpoints, and so forth. And I'll briefly touch on the Keytruda for MSI-high solid tumors, the tissue agnostic indication, as well as the Foundation One companion diagnostic next-gen sequencing test that was approved under the OCE. And then I'll discuss some of the companion diagnostic requirements. It's quite challenging, so it's not as straightforward as one would hope. So I'll define some of the challenges that we see in rare tumors as well. And okay, so the Oncology Center for Excellence was authorized by the 21st Century Cures Act as part of the National Cancer Moonshot Program by then Vice President Joe Biden. This was established in January 2017, so we just celebrated our one-year anniversary. And the goal of creating this center was really to combine the expertise across three major centers, really largely coordinated by the Office of Hematology and Oncology Products, which lives in the Center for Drug Evaluation and Research, because everything we do, every product we look at has a hematology or an oncology indication, but there are drugs and devices that fall into one of two other centers, the Center for Biologics, which covers things like CAR T cells. We, even though antibodies are biologics, technically, in OHOP, we still handle that. And then the Center for Devices obviously handles the device aspect as well as the diagnostic tests and in vitro diagnostics. So this is just an organizational chart so you can get a sense of where we live. This is the Office of the Commissioner, and these are the 3 centers that I just mentioned. And we all fall under the umbrella of the Oncology Center for Excellence there. It's not a physical place, but it's a sort of a coordinating center. So one of the efforts that was undertaken with the creation of the OCE was a sort of more synthesized review process, especially when multiple centers were involved. So the way this works is that sponsors will submit applications to the appropriate center. If it's a device, it goes to devices. If it's a drug, it comes to us. And then a determination on expedited review is made. So if that expedited review, and I will go into that in a moment what that means, but if that's granted, then the OCE creates a team. It includes OHOP within CEDAR and staff from the relevant product center, and then the review is conducted. And this is the time at which an advisory committee can be consulted or a special government employee to weigh in on the application. And then the review gets sent back to the product center, the primary product center. They make the final determination in the context of the other disciplines reviews. So I mentioned expedited programs, and I just wanted to share this because I think that there's some uncertainty and maybe some miscommunication on what these mean. There are 3 designations within the expedited programs and 1 pathway, and I'll get to the pathway in a moment. So fast track designation, it's based on preclinical or clinical information, and it permits more frequent interaction with the review team. Breakthrough designation is based on preliminary clinical evidence, and that indicates that an agent might demonstrate substantial improvement on a clinically significant endpoint over available therapies. And when I say available therapies, these are drugs that fall under the regular approval pathway, not the accelerated approval pathway. So it's important to make that distinction. And what the breakthrough designation offers is intense guidance and organizational commitment from OCE leadership, so to ensure that the most efficient development is being undertaken. Priority review designation has to do with the time clock and of the review, and it provides for a shorter period of review. The application, which would normally take 8 months, which would normally take 12 months, would take 8 months for a new molecular entity, and if not a new molecular entity, it would the time to review would go from 10 months to 6 months. I will say that the vast majority of things that we do in our office, excuse me, are priority reviews. So the expedited, accelerated pathway, which is also part of the expedited program, is a pathway to marketing. And it is based on the demonstration of a benefit on a surrogate endpoint that is reasonably likely to predict clinical benefit and provides meaningful advantage over available therapy in a serious condition. So there's a lot in that sentence. The important aspects to be aware of are that surrogate endpoint is used in lieu of a measure of direct clinical benefit. So direct clinical benefit is either measured as survival, is something that prolongs survival, or makes patients feel better or function better. And typically, the vast majority of drugs that have been approved with direct clinical benefit in the oncology space have been with overall survival. The applicants do have an obligation to complete post-marketing trials in order to demonstrate a direct clinical benefit, and then their applications or their drug would be converted to a regular approval. So some examples of recent accelerated approvals are the Keytruda for the MSI-high patients, as well as Opdivo for patients with HCC in second line, and both of these were approved based on response rates and duration of response. So I wanted to outline some of OCE's efforts in addition to those expedited pathways, which again pre-existed the OCE. So we have certainly, I'm seeing the fruits of the outreach to the oncology community, and certainly feel like I've been reached out to. But we're interested in hearing perspectives from patients, patient advocacy groups, researchers, and excuse me, clinicians. So this is part of this goes to that effort. So thank you for that. Another effort that we have is in reviewing eligibility criteria. So not surprisingly, eligibility criteria have traditionally been very stringent and restrictive, and this has prevented patients from being able to enroll on clinical trials, but it also sort of confines the experience to very healthy or very well patients and may not be a true representation of what we see in the public once a drug is marketed. So there was actually a position paper that was put out last year by our office on reevaluating eligibility criteria, things like CNS metastases, organ dysfunction, what else? HIV status, and so forth, and the age criteria as well. In terms of seamless design and expansion cohorts, this is a potential design that could be explored. And what it consists of is if an agent has an impressive benefit in the phase 1 category, an expansion phase can be opened up, an expansion cohort, excuse me, can be opened up, and this could potentially contribute to approval. And when I say that, I'm thinking of ceritinib for non-small cell lung cancer, for ALK-positive non-small cell lung cancer, or Keytruda for refractory melanoma. So there is a precedent for this. I would have to say that these are very labor-intensive endeavors because they require that the design of the trial is very clearly stated, as well as the objectives, and it requires a great deal of input from the statistical analysis side because this has to be pre-specified. But it is an area of opportunity, and currently OCE leadership is recommending or is suggesting actually that drugs that fall under the breakthrough designation category would be the best candidates for this, for looking at seamless design. We're also interested in looking at novel endpoints. We know that overall survival is not always a realistic or feasible endpoint, especially in tumors with a very long survival or rare tumors in which we can't adequately power trials because of a limited population. And there's also a concern that preliminary evidence may cause a loss of equipoise, and a second clinical or additional long-term trial could not be undertaken. So I think I had mentioned with the MSI-high, the tissue agnostic indication response rate. Now, again, that was accelerated approval, but response rate and duration of response were the basis for that approval. So these are considerations for endpoints that we can use, as well as PROs, by the way, which I think Katie mentioned. But because it is a direct, that is a measure of a direct clinical benefit if a difference can be shown. So I think the important thing to recognize here, though, is that the magnitude of the benefit and the robustness of the data really play into what's into the appropriate use of this type of data and these type of endpoints. So moving on to real-world data, I think this is sort of in development. This is sort of in development, but the FDA does have a data transformation initiative and some collaborations with agencies like Flatiron, CancerLink, and Project DataSphere in terms of exploring this type of data, which can help inform long-term safety and chronic toxicity, and may also be able to play into the risk-benefit analysis when we're looking at an approval. The final thing that has been a big effort for the OCE is the patient-focused drug development. It's many years now. Colleagues from my office have been working on the patient-related outcomes, working with different agencies internationally and nationally, but specifically with the goal of identifying a rigorous method for using this type of data. They were part of the development of the PRO-CTCAE, and we're currently looking at how to use that type of data. Okay, so I already mentioned this, the MSI high tissue agnostic approval, again, this was approved under the cross-center approach. And I just wanted to point out here, because we are talking about companion diagnostics, this was an accelerated approval, but in the majority of patients, a laboratory-developed test was used. And a laboratory-developed test is typically a single lab that has a test. And because this was a composite of multiple trials, of patients from multiple trials, there was no companion diagnostic that was co-developed here. Sorry. Now I want to talk briefly about Foundation One. The Foundation One companion diagnostic test, as you all know, this is a next-gen sequencing based in vitro diagnostic, and it detects mutations in 324 genes, and it was approved as the companion diagnostic for 15 targeted therapies in 5 cancers, specifically non-small cell lung, melanoma, breast, colorectal, and ovarian cancers. It also was approved using the cross-center approach. This was a notable approval for 2 reasons. First of all, it was the second in vitro diagnostic that was authorized under the parallel review program, wherein FDA and CMS simultaneously review the data, and obviously FDA for safety and efficacy, and CMS for coverage reasons. But the CMS part of it, because those happen simultaneously, it allowed faster access for patients. This companion diagnostic was also the first device under the pre-market approval process under the breakthrough device designation within CDRH. And so what that is, it was also mandated by the 21st Century Cures Act. For those of you who might know, there was a previous expedited access pathway, but the breakthrough device supersedes that. And it's really intended to get patients quick access to new devices and technologies. And it does allow for intensive guidance from CDRH, sort of similar to what the breakthrough designation does for drugs within CDER. Apparently, the way I understand it is that the review is prioritized so that the time, it may take a long time to review, or it may take whatever time it takes to review, but at least it's sort of first up on the list if it has that designation. So moving on to some of the background of how devices and diagnostics are approved. First of all, they're risk categorized. There are 3 risks, risk assessments, and these are based on the consequence of a wrong test. Is a patient going to be administered a therapy that they wouldn't qualify for because they had a false positive, and vice versa? Would they not be treated if they could have been? And in that type of scenario, this would be a high-risk device. So what a high-risk device is so what happens is that the high-risk device would acquire a pre-marketing authorization, and basically that allows them to put the FDA-approved stamp on it. This would require safety and effectiveness data. So things like cancer screening tools, predictive biomarkers, all fall under the high-risk category. Moderate risk category would receive a FDA clear designation, like a prognostic marker would be a good example. So most of what we're talking about here is going to fall into the high-risk category. So some of the elements, just to give you a flavor of what's required for an in vitro diagnostic submission, quite a list of things. Certainly the indication has to be included, device description, and pre-analytical data, where I think I want to highlight the analytical and the clinical validation of the assay in question. So the analytical validation is really the characterization of the test. Does the test measure the analyte that you think it does? So this is sensitivity, specificity, reproducibility, accuracy, and robustness. The clinical validation comes about from the clinical data and answers the question, does the test result correlate with the expected clinical condition, and how reliably? So it's going to depend on what the indication is. Is this a diagnostic? Is this monitoring for recurrence? Is this just a prognostic? It also depends on the diagnosis itself. So for a companion diagnostic, this is going to come from the successful drug trial. And there are some other things as well that are required, but some technical aspects. So oftentimes in clinical trials, the final test is not actually the one that was used in the clinical trial. And in this circumstance, bridging studies are required to link the clinical trial assay to the diagnostic. That's being reviewed. And I know that CDRH is a big advocate of saving all your samples and including a random subset of negatives. You need that negative control. And reanalyzing the data, the clinical trial data, in the context of the in vitro diagnostic results. So this also incorporates a statistical analysis plan that takes into account all the things that can go wrong, discordance, missing samples, and they recommend that sensitivity analysis be performed as well. So I think this is my second to last slide. Just some of the challenges in companion diagnostic development. These are well recognized. Things like the rarity of disease, the need for rapid results, inadequate specimens for validation or bridging studies, as well as the cost of developing a companion diagnostic. In talking with my colleagues from CDRH, they've encountered all of these issues, and they have tried to make allowances when appropriate for a laboratory-based test to be approved, for example, or a companion, or maybe an alternative strategy to obtaining samples for companion diagnostic approval. So the best advice that I can really recommend is to speak with them directly through a pre-submission program. This is the link to the guidance for how to go about doing that. But there are mechanisms, there is flexibility, and so it's worth discussing. So in conclusion, expedited programs exist to facilitate safe and effective drugs getting to the market. That's been the case for a long time. The OCE is sponsoring some new efforts to foster efficient development and review of drugs and devices, and contact CDRH. So with that, I'm finished. Thanks.
Thank you, Naomi, for giving us a peek behind the curtain as to how the FDA thinks, and we appreciate all the expedited programs that allow us to get drugs to our patients.